We'll continue on. Our next presenter is Marcel Proske. Proske.
No, Proske.
Proske.
Yeah, but it's fine.
I've known Marcel for 2.5 years, as you can tell. So actually, I first met Marcel when I went to Germany to get introduced to the Helmholtz Institute and Dr. Niesing's laboratory. And he's been working on pure alpha in one form or another, certainly since then. So this is your second conference, is that right? That he's presented at and has a lot of updates on the things that he has been doing as well. So please give a warm welcome to Marcel.
Can you hear me? Yeah, fine. So thank you, Dom, for the introduction. And I'm also happy to be here to present the work of Bettina Schmidt, because unfortunately she cannot be here. So Bettina Schmidt is working with a zebrafish as a model organism for your PURA syndrome here. And yeah, I will just lead you through a little bit what she's doing and what the zebrafish is actually like. So let us start with how the zebrafish looks actually. So I know the fish as under another name actually. You can see it in an aquarium or so. But here you see a female and a male version, which you can distinguish from the body shape. Like the male is more like a torpedo. The female has a rounder belly. The stripes are silver compared to the male, which are gold. They are like 3 to 5 centimeters in the body length, and they have a life expectancy of like 2 to 5 years. And which is quite nice for having it as a model organism is the reproduction rate. So they are like only need 3 months to be adult, and then you have can generate a next generation already, which is quite cool for a model organism because, as you heard already from Jen, the mice takes time, for example. So give you an impression about it's working perfectly, how those fishes are set up in the facility. They come in such boxes, and there's not only one box, you will see later. And in this video, you actually see how they breed, because then you have this S-shaped inlet there, so to modify the coast. And you have to put the inlet in because otherwise they will eat the hatching eggs, the fish. So it's not really nice, but they do it. That's why you have to separate them to generate the next generation. And okay, next one, yeah. In the facility, there's not only one box, there are hundreds of boxes, which can really, you can see here, and with hundreds and thousands of fish. And they are used to study now for the PURA syndrome. So you all will think about, okay, that's a fish, we are humans. Dirk already mentioned that they are not so close to us than mice, for example. But still, they are pretty close, which you can see in this cartoon. So we all are like vertebrates, which are like animals with a backbone, in principle. And you can see we are not so much different because we both have a brain, we have eyes, we have a heart, a lung, intestines, but we have our difference except for the outer shape actually. It's like they have a swim bladder, we have a lung, so they don't need a lung underwater, it doesn't make sense. So yeah, but the questions are now, how can we use those similarities you can see there actually? Or what we can use the zebrafish for. And for you guys, the most important question is, what can the zebrafish do for your kid? And there are like 2 answers. Dirk already mentioned some. That is like, we can investigate in the zebrafish, first of all, like defects in body plan. We can track developmental processes. And secondly, we can do a massive screen for a drug, for example, for epilepsy, which is pretty easy because they are in water. So you can just give a drop of the drug to the water. They will take it up with the gills, and that's it. Then you can see if it works or not. But I will come to that point later. So how does it help us to understand what's going on during development? Because still it's a fish. But if you check the embryo, there are some stages where all the embryos are like quite similar to each other. In this case, they are the same. In this stage, because you see there's already a part which develops later to the forebrain. Yeah, here. For the forebrain, there's a part for the eye, for the midbrain, and the hindbrain, for example. And that's for all of us the same in that stage. So, but the biggest advantage here is that those embryos are actually transparent. So you can look inside and see what's going on. Oh, sorry, that was too fast. And therefore we can track the development and understand maybe some malfunctions which are happening during developmental. In this video, I hope you can see it, that's just an impression what we can actually do just by looking the embryo under a microscope, because you can already see the bloodstream of such a fish. But that's not everything, because we can even go further and we can stain whole organs to track the development of them, such as motor neurons. Looks quite cool and similar to the mice Jen presented with the GFP glowing. But we can also go further and stain a whole organ like the heart of the fish, and of course also the whole blood vessels. And that will help us like to see what is going on in the fish and what is going wrong actually, because you can see. And the third one is, when does it go wrong? Because that's also a very important question. When does it start to go wrong? And we can use the fish also to modify. So we can now check like the normal fish you buy in the zoo, or you can edit it with this gene scissors, which is like, I think you are familiar or heard of the CRISPR-Cas9 system. And since those are eggs, you can modify the fertilized egg in the one cell stadium, and then you can introduce mutations and everything you want. And after 2 months, you will have the fish. You can mate it with another one, and you will get a cell line or embryos which have this mutation you wanted to introduce. That's quite an advantage, because then you can compare it actually to the healthy ones. So you see the differences. How is that done? So in the next pictures you see Bettina sitting on a microscope actually, and she's handling some wheels and a tool actually, which is here. You can see in this picture there are, you can see the mouse. There are the eggs laying in a row, those white dots, and the tool she is handling with those wheels here is like a really sharp and thin needle. And with that needle, she goes into those eggs, fertilized eggs, and then you can see how it is like injecting this CRISPR-Cas component just to modify the eggs and the fish. And that's how we can track then developmental differences and see in which kind of, we can introduce each patient mutation we found and see what are the differences. Secondly, I told you that we can use the zebrafish as a search model for drugs for epilepsy, which is quite important because as far as I know and I saw that kids have a lot of seizures mainly, and that's a really reduction of the life quality. So it's important to find something which can help, because there are no drugs at the moment which are helping there so far. And yeah, in the embryo here, again, they are super small, as you can see, and transparent. So you can do it in a big screen. And you just take 2 fishes who have the disease, and they mate them, so you will get eggs, and later the embryonic status. And you can add to the water those so-called compounds. Those are those FDA-approved drugs to test for epilepsy. And you do that in like a 96 well format. And there's an automated system which like observes and tracks everything of those larvae. And you can see which of those drugs actually rescues or like cure the not cure, but like to get off the symptoms of the epilepsy. And then when you get one of those, you can further test it in different model organisms. Like then you can say, okay, we found it in the zebrafish, that drug helps. And then we can give it, for example, to Jen, and she's doing in the mice model to get more closer to the human. So what you guys should take home now is that there are like various tools to actually track and investigate developmental disorders by just checking even on motor neurons, heart, and blood vessels. We can do a massive drug screen, which is really easy because you just put one drop to the water, and it's not like you have to inject like an animal with a syringe with the drug and see what's happening. It's just in the water, they take it up easily with the gills. So you can screen a lot of them because they are like fast in reproduction. They are small, so you have this 96 wall plates, and that's quite a big advantage. And the third one will be that you can introduce those mutations you want to have, and you can compare it actually to the healthy one and see if there are differences between in development or in behavior and so on. So that's the end of my talk. So thank you for your attention. And if there are any questions, just ask me.
Zebrafish are pretty cool, and that's again a new model that was just instituted late 18, I believe. Is that right?
Yeah.
So it's very exciting to see current big science, for our kids. So Marcel, thank you very much.
You're welcome.
Wonderful pleasure to have you here. Thank you.
Thank you. The pleasure's all mine.
All right. And Lena Molitor is next. Lena and Marcel work in the same lab. I also met Lena 2.5 years ago. Yeah, she's, yes. So Lena has I learned something new about Lena every time I meet her. And she's studied in 2 different universities in China, right? Yeah, China. In China alone, as well as working on her PhD with the Niesing lab in Munich. So, Lena, thank you very much for being here.
Yeah, hi everyone. Can you hear me? Yes, nice. Hey, I'm Lena. Thanks for the nice introduction. As Dom already said, I'm doing my PhD in Dirk's lab. I really like the sound of the name in English. Okay, so I'll be talking about what we are doing in the Niesing lab, because as you heard, the zebrafish is kind of like outsourced. Yeah, and I called it Grasp PURA because that's what we want to do. We want to understand what's happening. Okay, here we go. There's been quite a lot of introduction today. I still have an introduction slide because I thought it might be always good to repeat stuff, right? So we have the PURA protein. Protein is PURA, the protein, basically PUR alpha, contains 3 PUR repeats. And if you look at the structure, they're here labeled in like green, blue, and white. And green and blue together form like a domain. And a domain is nothing else but just a structure where those two, the green and the blue, interact. And Dirk always liked to call that like a molecular handshake because they interact something like this. So, and then there's a third part of the protein, that's the third domain, and that's in white. And this interacts with another PUR alpha protein. So they form something that is called a dimer. They like each other, and they come together, a homodimer, because it's PUR alpha and PUR alpha set 2 times the same protein. It's actually nice to remember that it's called a homodimer because it's the same protein coming together. And one thing that's really relevant for the stuff I will be talking about is PUR alpha is known to interact with DNA and RNA. Okay, so Dirk talked a lot about DNA, which is, again, the PURA logo. You remember DNA on the logo, and about the protein, PUR alpha. But he kinda, probably purposefully, left out that there's a step in between. So DNA doesn't right away form a protein if it's read up. So you can think of DNA as like a long-term information storage, and then it's decoded into a less long-term information storage, and then it's decoded to a protein, which is basically the active form of the information that does stuff. And the transition form in between is called RNA. And RNA is very similar to DNA, just it's less stable. That's why our information is stored in DNA, because it's very stable. RNA is then the version that's processed. And I'll be talking a bit about RNA because we know that PUR alpha, the protein, does bind to several RNAs that hang around in the cell. But we don't really know why or what's the function of this. And we really want to have the bigger picture, so we really want to know that as well. So basically, we know that PUR alpha interacts with a lot of things in the cell. Different other proteins with itself, which is also the homodimer I was talking about. It interacts with DNA, and it interacts with RNA. And I tried to list this, and we have the DNA interactions. There's a technique called ChIP-seq, where you can analyze a protein that interacts with DNA. But we didn't do this yet. This is like a work in progress. And then there's like a strategy to analyze protein interactions, and Marcel does this mostly. And the technique we're using there is called BioID, but I'm also not talking about this. I will be talking about the one in the middle that's called iClip. And I kind of see that it's not really intuitive that I marked the ones that I won't be talking about. But I thought, let's just do something different for once. So the technique I'll try to introduce to you is called iClip. And I've been working on this for like a little over a year. It's really fun. If you do not understand anything of this technique, I didn't understand the technique in the beginning either at all. So if you have any questions, please get back and ask. And ideally, we want to analyze this technique during the differentiation of cells, meaning we want to see what RNA does our protein bind to in stem cell state, what RNA does it bind to during development, what RNA does it bind to in neuronal cells, and so on. Because we really think that this process of binding to RNA regulates the development of neuronal cells. But we don't know. Okay. Technique. Just so you see how exciting science is. So in A, you basically see the initial process of this protocol, which is like so the entire protocol is a proc 60-step protocol. It is quite complex in my opinion, but okay, I just break it down to 3 steps. So you have the cells in a Petri dish. I always culture cells in a Petri dish. And you put them under UV light. And what happens under the UV light is that your protein of interest, in our case that's PUR alpha, is connected to the RNA molecule that it binds to. But it's cross-linked, so it's like coupled together. And then they stick together. And this is like a snapshot, because at this moment, this RNA was bound to the protein and we snapshot it because then it's cross-linked. And what we then do next is we have a process called Immuno Precipitation, but it's a complex word for an easy process because we have one protein, another protein that directly recognizes PUR alpha, and therefore we can, out of the entire cell, we can pick only PUR alpha. So we can get of an entire cell, we pick out only PUR alpha, because that's the only thing that interests us in this case. But since we cross-linked it before, we also get the RNA that PUR alpha was bound to. So we get both. And what we then do is, and that's really nice in modern day science, we can get the RNA, process it a little, and then we can sequence it. And then we see over the whole genome, where did our protein, PUR alpha, bind to the RNA? What RNA does it bind to? In the entire cell, we can know what RNA does our protein bind to. This is quite, in my opinion, it's quite crazy. I hope I didn't lose you. And what you get out of this is something like this. This is not PUR alpha data, I have to say, but what you get out of this is something like this. And the one in, what is it, like violet, would be the iClip data. And these peaks show you what are, on a whole genome level, what RNAs are bound to PUR alpha. In this case, it's not PUR alpha, but that's like the idea. And you can align this to a so-called ref-seq. They write there ref-seq, right? And ref-seq is the whole transcriptome, meaning all RNAs in the cell, and you can see the alignment to what PURA has bound. And then we can analyze this data. This is huge amounts of data, obviously, by bioinformatics, and we can figure out what does it bind to. And then we see, because we know some RNAs are relevant for this process, and some are relevant for this process. And then we can like pinpoint where is PUR alpha relevant, in what process. And that's the idea of the project. Okay, you can ask questions later. Good. Because now I'm getting to the second part of what we do, and it's very different. It's not specifically protein and RNA interactions, it's more modeling the entire disease. And I think it might be a little easier to understand. And for the people that were in Cambridge last year, it's very similar to what I presented there. So, first of all, the person right on top, that's Aona. She's a really good friend of mine. It's very random. No, she works in the Stem Cell Institute, and she works together with me. And it's, everything that I'm showing now wouldn't work without her, because she has a lot of expertise and putting a lot of effort into this. So, what we want to do, as everybody else already presented to you, we want to model the disease, PURA syndrome. But we want to model it in human cells. Because, I mean, that's genetically as close as you can get, because it's the same organism. Still, it has the limitation that we cannot have tiny humans in Petri dishes. So we kind of have to find a way around this, and therefore we culture only cells. No organisms, nothing, it's just a cell. So a cell is basically something that you only see in a microscope because it's so tiny, and you let it grow in a Petri dish at like 5% CO2 and 37 degrees. So like in our body, and you put it in an incubator, and it grows. Sometimes you have to change media, that's it. It's nice. And the cool part is there's a technology, and I listed this here because it says PURA iPSCs. There's a technology where you can take cells from the human body, skin cells. You take them, you put them in such a Petri dish. They will grow. And then you add some magic chemicals, and they become pluripotent stem cells, which is fantastic. This technique got a Nobel Prize, like in 2012. Shinya Yamanaka is the guy. If you want to read about him, he's like a spectacular researcher. And he generated for the first time from a differentiated cell a stem cell. This is insane, I think. No? Okay. Right, and then we have these so-called iPSCs, which are pluripotent stem cells, and they have the capacity to differentiate into all the cells in the human body. Again, taken from a cell from our body. So we take them back to the level where they can do everything, and then they can go down again to all cell types. So we can take a skin cell, make it a stem cell, and then generate a neuron from it. That's possible. Like, it's crazy, I know. Right, so what we want to do, because we want to have like a controlled system, we always need a control cell line. So I listed, on top you can see that there's a healthy individual. We take a skin punch, we generate those stem cells, and then by using CRISPR-Cas, I think it's been pretty much in all the newspapers lately, we modify the cells in order to not have functional PURA alpha in the cells anymore. And then we have, compared to this, the healthy control cell line that has functional PURA alpha. And then, as you may see, if we then have the stem cells, we can differentiate towards neuronal cells, for example, but we could also do bone cells or all the other cell types. And we can see during the process of generating these specific subtypes, what goes wrong in the process in real time in our tiny Petri dishes. And that's really an outstanding technology. And the other controlled setup would be that we have PURA patient skin cells. We take them, make iPSCs, and we did that, and we then repair by CRISPR-Cas the region that has the mutation in the PURA gene, and then we would have to see that these cells behave like our control cell line initially. And this would be another comparison. So if we have those 4 cell lines, we can basically very nicely analyze the process of differentiation or generating neuronal cells and so on in PURA syndrome. So that's the idea. So we generated a PURA patient iPS cell line with a phenylalanine 233 deletion. We got a skin punch sample from a patient. We reprogrammed the cells to become iPSCs. And now we have these cells in culture, and we can look at them. And that's pretty cool. First of all, we have to validate that these cells are actually stem cells that we generated. And we do this by coloring them, just like kids, you know, we color everything. In this case, it's so-called pluripotency markers. So if you see a color for these markers, that means that they're kind of pluripotent. And we, in this case, used Lin28, Nanoc, OCT4, and SOX2. And all of them are positive, means our cells are fine. So they are actually pluripotent cells from a PURA patient. We generated them. So we're good to go, basically. And I have like some examples that what we want to do or what we already did. So we want to differentiate these stem cells to neuronal cells first, but as I said, we can do all the other cells if there's like a clear indication that this would make sense. So for example, endocrine cells might make sense, stuff like that. But right now we do astrocytes and neurons, which is both cells in the brain. And for example, and they all go through a state called neural progenitor cells, NPCs. And for this example, I picked out one Western blot. Western blot is also a very fun technology. But basically, as Jen already mentioned, it's looking at a protein that you want to see. And in this case, you always have a control. So on the left side, you can see the control, which is tubulin. It's like a protein that is in all the cells in a similar amount, just to normalize our protein of interest. And on the right side, you can see PURA alpha. And what you see, so clone 4 and clone 11 are patient clones, and you clearly see that there is less PURA alpha, right? The band you see at approx the number 41 is the band for PURA alpha. You can see in the wild type, wild type is always the healthy cell. Well, we call it wild type. It's a weird word for it, but yeah. And then you see that there's a lot of PURA alpha in the wild type, which is on the far right. And in the knockout, there's nothing. The knockout is the one that I generated, where it's no PURA alpha. And in the patient cell, you can see that there's something in between, like a medium amount of PURA alpha, which makes sense because all kids have only the mutation in one of the 2 alleles. So there should be some PURA alpha left. So we can do this kind of analysis. And I have just an outlook or like an overview of what we already did. Everything that has the green mark is already done. Everything that is red is supposed to be done by my master's student. Exactly. So we did some different differentiations. I already, like for example, with the astrocyte, you see that the differentiation takes 50 days. So if you imagine you always have to do biological replicates, you have to do this like 3 times in order to say something. So you have to do 3 times 50 days. It takes some time. It takes some time. But we're getting there. And I'm really excited that we now have the tools at hand so we can start doing a lot of experiments there. With that, I would actually already like to acknowledge people. And since Dirk had such a nice picture of me with the wine, I decided to put also a nice picture of him. I'll just let it sink in a little bit. That was when a paper of my colleague was accepted, and we were really excited because it was in Nature Communications, and in science, like Nature papers are always nice. Anyways, we were excited. Dirk was obviously excited. He thought he was hot. Finally, okay, acknowledging people actually. Yeah. I mean, all this is like always a huge team effort. There is nothing that anybody by himself or herself does. It's a team effort, and the lab is fantastic. It's a real pleasure to work there. I think if you ever have anybody who wants to do a PhD or a master thesis, like, let us know. Let us know. We always are looking for motivated people to join our lab, and it's a fun atmosphere. You just have to come to Munich, but it's a decent place to live, I think. Yeah, so basically I want to thank Dirk, obviously, who does all the general like instructions, helps us with all the project and so on, and Marcel, who's like who you already saw, who's working very closely with me, and Verena and Sabrina are 2 students of mine. Verena was a bachelor student. Sabrina is now a master student who starts doing her PhD on the PURA syndrome in our lab now. Which is very exciting, I think. Annika and Jana are former alumni of the lab, but they helped setting up a lot of things. The Drucker lab, Micha Drucker, who was here, and Iona, as I already mentioned. The Dorman lab, they kind of help us stain and look at poor alpha and so on as well. Koenig lab, that's the lab that we are collaborating for the sequencing of the RNA that I showed in the beginning. They're a huge help with that, and obviously, I want to thank the foundation. It's fantastic to be here. It's fantastic to have these events. And for us, I think it's super relevant to see why we are doing this and how this affects all of you here. And yeah, it's just great. And then finally, we're on Twitter. So if you want to like see us continuously, just go for the Niesing Labs on Twitter and follow us. We try to update regularly. And then the picture is our lab on top of Zugspitze, the highest mountain in Germany. It's nice. Thank you very much.